Hello students, this is Dr. Sujata Magdum, Assistant Professor in Zoology, KTHM College, Nashik. Today I'll be dealing with Biological Chemistry for TY BSc students, and the topic of discussion is colloid and their important properties. Right? Earlier we have studied about the various different kinds of bonds which are present into the nature, and that results in holding the different atoms and molecules together, as a result of which large number of biomolecules they are seen clubbed together, giving rise to certain structural entity. Now, one of the structural important entity which is seen into nature are the colloidal substances, and that we are going to study. Colloid is a Greek term and is defined as glue-like, or it means glue-like. The word from Greek is kolla, and these colloidal particles or colloid, they are consisting of two important components, that is the dispersed particles and the dispersion medium. Dispersed particles, they are always seen into the dispersion medium. Now this dispersion medium could be any solid, liquid or gas. You have to remember that gas in gas, that means the dispersion particle can never be a gaseous molecule getting freely dispersed into the gaseous medium once again. Thus it means that either the solid can get dispersed into the gas, solid can get dispersed into the liquid, liquid can get dispersed into the solid, or liquid can get dispersed into the gas. As a result of which, the entity what we get is going to be defined as the colloidal particle. Now, as I am stating two important terms, that is the dispersed particle and dispersion medium, we need to understand what exactly they mean. Dispersed particles, these are also called as discontinuous phase of molecules, which measures approximately 0.1 micron to 1000 micron. And these are never seen with the help of optical microscope, but can be confirmed with the help of ultra microscope or electron microscope. These dispersed particles, they are always seen mingled with or freely suspended into the dispersion medium, which, as I said little 2 minutes back, that the dispersion medium has to be either solid, liquid or the gas. Hence, these dispersion medium, they are said to be continuous phase. Now, why they are called to be continuous phase? Because you can see their occurrence, their you can say dispersion continuously, whereas the dispersed particle, because they are aggregates, they are never seen into the homogeneous state. Rather, they will be seen settling down towards the lower side. And here you can see the dispersion medium is been shown by orange. The dark orange shows the colloidal state. The liquid state is been shown with the help of light orange color. And the green balls, what you can see within this orange marking, is defining the dispersed phase of the colloid. Now these particles, that is the dispersed particles, are never seen getting passed through the filter paper, but they can be seen passing through the semi-permeable membrane. That means their size is so in between. Let me put it again, that these dispersed particles, they can pass through the filter paper, but they cannot pass through the semi-permeable membrane. And centrifugation is possible, which results into settling down of these dispersed particles. Now, we'll try to understand the distinction between the true solution, colloidal solution and the suspension. Let me begin with the pictorial representation. There are three different boxes wherein the blue color, light blue color, indicates the medium, that is the dispersion medium, whereas the dark blue balls, they are representing the particles. You can see that in the first picture here, the particle size is very small and they are large in number. So this is what is seen is the condition with reference to the true solution. Let me now put it into the terms of biochemistry, wherein I can say the light blue color is defining the solvent, which could be either the liquid phase or it could be the gaseous conditions, whereas the small blue balls, they are defining the particulate nature, which could be of any atom, ions or molecules, which gives rise to formation of a true solution. Whereas when we come to the next picture, that is the colloidal solution, here you can see that the light blue part, which is defining the solvent, it holds large number of slightly larger round spheres, which are the particles. Obviously here we can say the distinction between the true solution and colloidal solution is with reference to the particle size. The particle size happens to be much more larger as compared to the particle size which we have observed into the true solution. Whereas in the third box you can see that there is light blue solvent, and this is holding once again very large particulate matter. Once again, if I compare the particulate matter amongst all these three, we can say that the suspension will have very large size of particulate matter. That forms the distinction between true solution, colloidal solution and suspension. Probably this is the only reason because of which these colloidal particles, they are not seen entering through the semi-permeable membrane. That is, the particle size, it matters a lot because of its intermediate nature. It cannot be seen moving across the semi-permeable membrane. Now let us try to define these colloids. Colloids are the polyphasic systems where at least one dimension of the dispersion phase measures between 10 to 100 angstroms to few micrometers, right? Now let's move ahead. Now these are the different properties what we need to understand, which will help us to define the distinction between true solution, colloidal solution and the suspension. If we consider particle size, in case of true solution it is less than 1 nanometer, whereas in colloidal it is in between 1 to 100 nanometers, whereas in case of suspension the particle size is more large, and hence it is always observed more than 100 nanometers in size. Because of this variation in the size, there is transparency, or you can say the appearance of the solution happens to be very clear with reference to the true solution, whereas in case of the colloid and the suspension, it appears to be cloudy. Now, this feature of clear solution or cloudy solution will help us to define whether the solution is homogeneous or heterogeneous in nature, and therefore the third property, that is homogeneity, we can say true solutions, they are homogeneous in nature, whereas colloidal as well as suspensions, they are heterogeneous in nature. In the same manner, because we are defining the homogeneity, now we can talk with reference to the transparency of the solutions. True solutions can be said to be transparent because once again it is related to the size of the particles which are seen into the dispersion medium, whereas heterogeneous colloidal or the suspension particle, because of their larger size, they appear to be translucent with reference to colloids, whereas opaque with reference to suspension. The difference between translucent colloid and suspension is once again due to the size of the particle. In colloid, as I said, the particle size is slightly larger as compared to the true solution, which measures between 1 to 100 nanometers. It gives the translucent nature to the solution. Whereas in case of suspension, the particle size happens to be very large, and that has resulted into giving the opaque nature to the solution. Now, if we try to separate these solutions, we can say that true solutions, in this true solution, both the dispersion medium and the dispersion particles, they cannot be easily separated. Whereas in case of colloid as well as suspension, they can be easily separated. Now, this separation can result with the help of centrifugation. Now, precipitation reaction, if we try to understand, we can say that in true solution, the precipitation is not possible unless and until the solution is saturated. If it is not, then the process of centrifugation will not lead to precipitation. Whereas in case of colloids as well as in case of suspension, there is chances of precipitation because of the larger size of these particles. Because of this precipitation as well as size of the particle, we can talk about the filtration property, that is filtrability. Now here we can say true solutions, they can easily pass through the filter paper and not the semi-permeable membrane. Whereas the colloidal particles, they can pass through the filter paper but they cannot pass through the semi-permeable membrane. The reason why it cannot pass through semi-permeable reason goes to the particle size. That means because there is large particle size, the property of passing through the semi-permeable membrane becomes little impossible. The same property goes true even with the suspension. As the particle size is very large, these solutions will not help in separating the dispersion particles from that of the dispersion medium, and therefore we can say that there these will not be able to see passing through the filter paper, neither through the semi-permeable membrane. Now let us talk about the physical properties. Now the first property which I am going to define is the heterogeneity. Heterogeneity defines the dispersion phase and dispersion medium we are going to consider. Now what does it mean, dispersion phase and dispersion medium? It means that the colloidal solutions are, because they are heterogeneous in nature, there are these two different phases, and these phases are formed because of the particles which are freely seen suspended into the medium, that is gas or the liquid, that is forming the dispersion medium. Now these particles which are the dispersed phase particles, they are of various sizes and shapes, and these various sizes and shape, they are responsible for imparting color to the solution. The best example is gold particles, which are spherical in nature. They give red coloration to the solution, whereas if the particles are of disc-like, then the color of the solution what we get is the blue coloration. So that defines the heterogeneity. So what exactly you need to understand here is, why are we calling it heterogeneous? Because the particles which are present, they are of different shapes and sizes, right? Then visibility. Now this visibility, although colloidal solutions are heterogeneous in nature, yet the dispersed particle present in them are not visible to the naked eyes, as I mentioned in the earlier slide, and they appear to be homogeneous. But actually speaking, these colloidal particles are too small to be seen with the help of naked eyes, right? But they can be viewed with the help of electron microscope, or you can say ultra microscope. Then the surface area of these particles are very small. You can see here I have tried to define one of the single small particle. Now this entire sphere is a surface, um, showing the surface area of the molecule. The solid center portion with the solid color, you can say, it is defining the center of the particle, that is the core of the particle, which contains large number of you can say protons, right? Now here what I have wanted to define is, the particle size is very small, and because its size is very small, it has a larger surface area, and this large surface area, it is charged area. Now here I have shown the negative charges on both the molecules, but it could be either positive, it could be negative, any one could be seen. That means either positive charge or the negative charge can be obviously observed, and these charges, they are responsible for making them soluble into the solvent. Okay, and these small spheres, that is the dispersed particles, they are forming effective catalyst because of presence of these charges, right? Then surface charges, the interior part which is neutral in nature. Now these interior part which are neutral in nature, and the surface area which shows presence of the charges leads to stability of the colloid. Now that is because they are not tending to attract each other, because all the molecules, all the particles rather, not molecules, all the particles here, they show the similar charges, and like charges they repel each other. So there is no aggregation and they tend to show the homogeneous nature or suspension, right? Then let us talk about the solubility. Solubility refers to their free mingling. Now, these solubility can be defined with terms of lipophilic nature or lyophobic nature. Lyophilic means these are readily dispersed by the solvent and don't precipitate very easily, and as a result of which the solution what we get is called as a lyophilic solution. Once again, this part I'll be discussing later in the next lecture. These solutions, they are reversible in nature. Examples of lyophilic solutions are gum, gelatin, starch and protein. Lyophobic are those solutions which don't disperse in the solvent, and as a result of which they can be easily precipitated. And because of this precipitation property, we can say the solutions are irreversible. Example of lyophobic solutions are solutions of metals, metal hydroxides or metal sulfides. Stability is the next point. Lyophilic solutions in general and lyophobic solutions in absence of substantial concentrations, they are truly stable. Color of the solution, it totally depends on the colloidal particle, what we have discussed in the earlier stage. Now let us talk about the optical property. Now, what do you understand by optical property? When an when an intense converging beam of light is passed through a colloidal solution kept in a dark, the path of the beam gets illuminated with a bluish light. This phenomena is called as the Tyndall effect, and the illumination part is called as the Tyndall concentration. Tyndall path. This phenomena was first observed by Tyndall in 1869, and the effect of Tyndall is due to the scattering of light by the colloidal particles. Since the dimension of colloidal particles are comparable to the wavelength of ultraviolet and visible reaction, they scatter these radiations and get illuminated, right? Now, this Tyndall effect was observed that the zone of scattered light is much more larger than the particle size itself. This is why colloidal particles look like bright spots when viewed with the microscope at the right angles to the beam of light, and this is shown into the diagram. Thus, Tyndall effect may be defined as scattering of light by colloidal particles present in the colloidal solution, right? This Tyndall effect is not exhibited by the true solutions. This is because the particles, that is ions or molecules present in the true solutions, they are very small and therefore scattering is impossible. Now the last part with reference to this Tyndall effect is, this Tyndall effect can be used to distinguish a colloidal solution from the other solutions like true solutions or the suspension solution. The phenomena has been used to devise an instrument known as ultramicroscope. This is what I have told you, right? Now, the mechanical property. The mechanical property of colloidal solution can be defined with the help of Brownian movement. Colloidal particles present in colloidal solution, they exhibit a very important property, and this property is called as the Brownian property. When a colloidal solution is viewed under the ultramicroscope, the colloidal particles are seen to be moving continuously in a zigzag manner. This property was discovered by the botanist, that is Robert Brown, in 1827, and he observed that the pollen grains suspended into the water exhibit random zigzag motion. After the name of the discoverer, the property is been named as the Brownian property, which is commonly observed into all the colloidal solutions. Now, the next, that is the electrical property. As I mentioned that these particles, they are charged particles. So these charged particles, they are responsible for giving the charge to the solution. Now, in a particular colloidal solution, all the colloidal particles may carry the same type of charge, while the dispersion medium has an equal but opposite charge. Thus, charge on colloidal particle is balanced by the dispersion medium charges, and the solution as a whole is becoming electrically neutral, right? Now, from where these charges are seen originated? There are several views regarding origin of charges on the colloidal particles. According to some, it is seen due to dissociation of adsorbed molecular electrolytes. Now, what do you understand by this? The colloidal particles have a strong tendency to absorb reactants or the product molecule. The molecule thus absorbed on the surface of colloidal particle may undergo dissociation or ionization and may thus give the charges. For example, during preparation of sulfide solutions, that is H2S, in chemistry lab you might have done, this H2S molecule has adsorbed, undergoes ionization and releases H plus ions into the medium. Consequently, colloidal particles are left with all the negative charges. Now, the second important property, second important view, which help us to understand the origin of charges is dissociation of molecules forming the colloidal aggregation. Now, here the molecules are responsible for formation of aggregates of colloidal dimensions, and they by themselves either undergo the process called as dissociation or ionization, and this results into development of charges on the colloidal particle, and as a result of which they become aggregated, right? For example, in case of soap molecules, dissociation of this soap molecule will give rise to two different types of component, one containing RCOO negative and the sodium ions. So RCOO negative ions will come together to form the micelle, which may carry a negative charge, right? This you will be understanding when we'll be talking about in detail the different types of colloidal solutions in the next lecture. The third important property with reference to electric charge origin on colloidal particle is due to preferential absorption of ions from the solution. Now, this colloidal particle has a tendency to preferentially adsorb or get adhered to a particular type of ion from the solution. A colloidal particle usually adsorbs those ions which are in excess and are common to their own lattices. For example, if I quote an example of ferric oxide solution, in that the hydrolysis of ferric chloride in warm water will result into formation of FeOH3, and this will give rise to Fe3 plus ions into the solution. This is because Fe3 plus ions are common to the lattice of FeOH whole thrice particles, and Fe3 plus ion thus adsorbed will impart positive charge to the colloidal particles present into the solution, right? Then the next important property is these colloidal particles, they can be separated with the help of electrophoresis. Now, what is electrophoresis? It is a phenomena by which the movement of particle is towards the opposite electrodes, and this is because of the influence of electric charges under the electric field. So because these particulate matters or the dispersion particles, they are charged particles, it is very easy for us to separate these charged particles with the help of electrophoresis. Likewise, if we need to understand the electric osmosis, we can say osmosis, you all know it is the movement of particles through semi-permeable membrane. But the same phenomena, when it is under the influence of electric charge, it can be called as an electric osmosis. So these colloidal particles, they show or they exhibit the electroosmosis phenomena, wherein because of their charged nature, they can be made pass through the semi-permeable membrane. Now, how this is significant over here? If you recall, I have stated that these colloidal particles or the dispersed particles, they cannot pass through the semi-permeable membrane by themselves. But by the influence of electric charges, these molecules, they can be separated or they can be made to move through the semi-permeable membrane under the influence of electric charges, and so they exhibit electric osmosis. The last important property of these colloidal particles is they exhibit coagulation or flocculation. Now, what is coagulation or flocculation? It is when the particles are seen aggregated together, and which gives rise to formation of a sufficient large component which can be easily precipitated. Now, such particles are defined as floccules or they are called as coagulate. Now, these coagulates or floccules is possible only because all the molecules which we have considered, they show the charged nature, and therefore we can say that these colloids, they exhibit coagulation or flocculation. I hope all the properties as well as the introduction to colloid is clear to you. In the next class, we'll be trying to understand the different types of colloidal solutions, right? Till then, stay home, stay safe. Thank you. Thank you so much.